Welcome to today's Gen webinar, which is entitled Genome Editing Tools Beyond Discovery. Our webinar today was made possible through sponsorship from Aldevron. I'll be your host today. I'm Jeff Bogaliskas, technical editor for Gen, which has been the leading source of information on the newest tools and technologies within the life sciences industry for more than 40 years. Well, 10 years after the seminal CRISPR gene editing paper by Dowden and Charpentier, the therapeutic potential of this tool has been realized and already begun to reshape some patients' lives. However, developing new CRISPR-Cas9 based gene editing tools will enhance innovative therapeutic products as well as the pathway to clinical success. Yet, as with any novel innovative technology, obstacles often crop up and need to be addressed to streamline workflows, ensure product integrity, and increase patient safety. Now, for current genome editing tools, the main obstacles that investigators need to address are things like high error rates, toxicity from production impurities, and strict regulatory concerns. Therefore, offering the optimal foundation for gene-to-protein services, from process development optimization to scale-up manufacturing, is critical to accelerating the therapeutic program to success. Tools such as Nanoplasmid next-generation vector technology provides that fundamental platform for extending and increasing expression, lowering transfection toxicity, and eliminating the risk of transferred antibiotic resistance to patients, something we're going to hear a lot more about in just a few moments. But first, let's take a moment to meet our presenters for today's event. It's my pleasure to welcome Max Sellman, Product Manager for Gene Editing at Aldevron. Max will briefly introduce us to Aldevron and the products and services that they offer, which can aid your gene editing research. He'll then turn the presentation over to our main speaker for today's event, Dr. Jim Williams, who is the Chief Scientific Officer and Vice President for Research and Development at Nature Technology Corporation, an Aldevron company based in Lincoln, Nebraska, that is a leading developer and provider of non-viral vectors, technologies and services for advancing gene and cell therapy and immunotherapy. Now, Jim is a specialist in non-viral expression vector systems with extensive experience in biological development. His expertise includes molecular biology, regulatory affairs, quality, molecular genetics, fermentation, and protein expression systems. Jim is the inventor of vector innovations, including the Nanoplasmidᴶ, and holds 31 U.S. issued patents with multiple foreign equivalents, along with numerous publication credits. And today, Jim is going to discuss features and data on using nanoplasmids as HDR template paired with Aldevron Cas9 nucleases, delivering premier tools for gene editing in CAR-T applications. Now, before we get the webinar underway, I want to remind the audience that we'd love to hear from you in our Q&A session that will begin right after the presentation ends. Max and Jim will be on hand to answer as many of your questions as they can. So simply type your question into the Q&A box and hit submit. Now, you can do this at any time during the presentation. You don't have to wait until the end. So if a thought pops in your head about Max or Jim's presentation, just go ahead and type it in, and we're going to try to address it in the Q&A session. Okay, with all that said, I'm going to turn things over to Max to get the webinar started.
Hi, everybody. Thanks so much for joining us. My name is Max Sellman. I'm the gene editing product manager here at Aldevron within our protein team, and super excited to talk with you today about some of the gene editing tools and services that Aldevron has developed and that we're using to serve the cell and gene therapy market. First, just a little bit about Aldevron for context for folks who might not know. We're a manufacturing company at heart. Been in business for almost 25 years. And what we like to say is we manufacture the entire central dogma of molecular biology. So whether that's plasmid DNA, mRNA, or protein, research grade through GMP, we serve clinical and commercial clients around the world with these high quality reagents and services associated with them. Our goal is always to be the supplier of choice for cell and gene therapy developers. When we're thinking about gene editing tools, Aldevron, for a little more than 5 years now, we've had a portfolio of gene editing proteins that are really intended to ease the transition into the clinic and then actually through clinical trials, as well as research grade bench work. And so when we look at our set of proteins that we have, we really think about trying to have a consistent approach to how we put these nucleases together. So really simple construct designs. You've got your various nucleases, which you see here in the different colors, and then a common construct design. So N-terminal and C-terminal nuclear localization signals to, of course, aid in transport into the nucleus. But apart from that, there's really no other features appended to these nucleases. We don't have affinity tags that we use in purification. There's no other markers or anything like that. There's a really simple construct design that's intended to kind of a streamlined regulatory approach and ease of use. And so we'll focus in on a few of these nucleases throughout the talk today, particularly kind of the ones on top. But these are available all as research grade tools. And additionally, we have a few that are available off the shelf as GMP catalog proteins. So that's a CGMP 21 CFR Part 211 manufacturing that we do here at Aldevron. And we've been manufacturing these products for a few years at GMP. And the idea that we had here is that you would have the same exact product that's available at research grade, also available at GMP. And so when you transition from the bench into your clinical programs or your preclinical work, you can expect equivalent performance from these nucleases. It's not better, it's not worse, it's the same because they're the same proteins ultimately derived from the same plasmid, expression plasmids, using the same manufacturing workflows and released with equivalent quality control testing panels. So, we've been doing this for a few years, and we're fortunate now to have a great body of stability data as well as drug master files available for your IND submissions. The idea here is, again, just really ease of sourcing, getting right into your clinical work. You don't have to source a custom batch of these proteins, and they're just available for for use pretty much immediately. The two that we have highlighted here on screen, which we'll spend a lot more time kind of looking at data. You've got a wild type SP Cas9 nuclease, which I think everybody here is likely familiar with. We also have a high fidelity nuclease that we manufacture in partnership with IDT, our sister opco at Danaher, which is SPI-Fi Cas9 nuclease. It's a high fidelity protein that has greatly reduced off-target effects. We'll get into that a little bit more when I start showing some data. Something that we're really excited to talk about is kind of our latest, greatest product, is what we're calling the Eureka-5 nuclease. So Aldevron has a licensing relationship with Inscripta, where we've brought in their MAD7 nuclease. And we've put this, like I said, into our existing construct design, and we plan to take this to GMP analogously to the two previous products that I mentioned, so as a catalog product. We're really excited about Eureka-5, this MAD7 protein. If you're not familiar, Eureka-5, this is a type 5 CRISPR nuclease, so it targets a T-rich PAM sequence, and it kind of behaves analogously to other type 5 nucleases that you might be familiar with. Very easy to get your hands on. You can contact us. We're going to launch this as a research grade product this month. And next year we will have it available at GMP. With purchase, you're able to use the product in your research or your clinical use without necessarily taking a license from Inscripta. But that's something that we're always glad to support and talk more about if you have interest in this Eureka-5 nuclease. As I mentioned earlier, Aldevron does manufacture what we call SPI-Fi Cas9. And this is a really exciting nuclease. We've had it for several years now, and this actually dates back. There's a paper that was published jointly by the team at IDT and Matt Porteus's lab at Stanford, where they characterized what IDT calls their hi-fi nuclease. And what's amazing here is that it's a single point mutation to the SP-Cas9 sequence, an arginine to an alanine, position 691. And the effect of that mutation is a near complete elimination in off-target effects. And so, as you, I'm sure, are aware, having off-target effects is an area of concern in the context of a clinical program for gene editing in humans. And so, when it comes to strategies for reducing off-target editing, having tools, having nucleases available that could very easily plug in and have the effect of reducing those off-target edits is a key tool in the toolbox. And so, in terms of performance, it's important to note, this protein is with that single point mutation from the wild type SP-Cas9. It is basically plug and play into existing wild type SP-Cas9 workflows. Uses the same guide RNAs interchangeably, can hit all the same targets that a wild type SP-Cas9 can hit, NGG, PAM. And here's some data that was actually generated from our partners at Vascugen, who edit iPS cells, just looking at hitting different targets with SpyFi-Cas9, trying to knock these targets down in iPS below their intended level for their application. What they were finding without doing very much optimization, that they were able to get really robust knockout using the SpyFi-Cas9, which of course is kind of your first benchmark of performance. Are we getting good editing efficiency? Are we getting the on-target results that we'd like to observe? And here would be an example of a client that's developing kind of towards therapeutic avenues, in this case for vascular disorders, and getting really robust gene editing. And so, it's just kind of exciting results that speak to the efficacy of this nuclease in terms of actually hitting the target that you intend to hit as a high fidelity nuclease. I know that's a concern with some of the earliest technologies in this space, was that they reduce off-target editing, but they don't do a great job of on-target editing. And so, that's kind of our first metric here, is do you get good, robust gene editing that's therapeutically relevant? And at least, in this data set, the answer there is absolutely. We actually have done some of our own work in conjunction with some collaborators. So in this case, we generated some data with the team at MaxCyte, who are, of course, experts in delivering reagents into cells. And here we were asking the question of, can we do multiplexed knockout in T cells? And so what we were doing here is reproducing data from a paper that came out of University of Pennsylvania in 2020. And what we were doing is we were doing a triple knockout. So 3 different guides targeting 2 receptors in T cells, the T cell receptor and the PD-1 ligand receptor. T cell receptor has an alpha chain and a beta chain. So that's 2 guides that you would target to knock out the T cell receptor and then the PD-1. And what you're seeing, as I kind of walk you through this data, on the left-hand side of the screen, you see kind of your pre-editing population in terms of cell by flow, looking at cell surface expression of these 2 receptors. Your middle set of data is when we target those guides to the receptors using a wild type RNP, so that SP-Cas9 from Aldevron, and you see really robust knockdown of expression of these 2 receptors. Notably, when you plug in the SpyFi-Cas9 nuclease, it's just a 1-for-1 substitution of SpyFi instead of the wild type Cas9, same guides, et cetera, same experimental conditions. You get that really equivalent robust knockdown. So, demonstrating here that SpyFi and wild type Cas9 perform really equivalently in terms of their on-target performance. So just to pause for a question, I mean, I know that this audience is, I think, well aware and well concerned about off-target effects and, focusing on the reduction of off-target effects. Aldevron, we're always curious to understand kind of the scope of your challenges with off-target editing. So we'd just love to ask the question to the audience here in your poll window. What sort of off-target editing are you seeing? Are you looking for off-target edits? And if so, at what level do you typically see? Appreciate your feedback here. Now, of course, if we're talking about a high fidelity nuclease, the off-target performance, of course, is important. And so, continuing on those experiments with MaxCyte, in reproducing that research from UPenn, looking at off-target sites that were predicted and trying to assess whether or not you could get that good reduction in off-target editing. And so what you're doing here is predicted off-target sites based on the sequence of the guide. And essentially we do our gene editing. We can do amplicon generation, and then sequencing at those sites, and ask the question of, what rate of off-target editing did we see in cells under these experimental conditions. And at some sites that we looked at, there wasn't a huge difference between the wild type Cas9 and the SPI5 Cas9. It's not always necessary to use a high fidelity nuclease in order to reduce off targets. I think we know that guide design, of course, plays a large role. But nonetheless, there are circumstances where you are going to see off-target editing, and you're going to see them at rates that are potentially concerning. And so at these two loci, we saw, when you used a wild type Cas9, something in the range of 4% or 2% of reads showing off-target gene editing. And simply by substituting in the SPI5 Cas9, the high fidelity variant, you see that value, that off-target value, drop from 4 or 2% down to background, down to equivalent levels of off-target reads that you would see in the samples that weren't electroporated at all. And so this is kind of a real-life example, real-life application of SPI5, demonstrating that the off-target effects can be greatly reduced simply by substituting one for one in this high fidelity nuclease instead of a wild type nuclease in this T cell application. So, we're very excited that, this nuclease has been around for several years, and, we're still seeing results like this. The more clients we talk to, the more experimental workflows that it's plugged into, it's a really consistent result to see this extreme reduction in off targets. So to the extent that that is something that is a challenge for you, this could be a solution that could help out. That's pretty low effort in order to arrive at. And we're very excited to have this nuclease on hand. It's something that we're, as I mentioned at the top, it's offered as a catalog product, research, and GMP. It's something that we're working into our ribonucleoprotein manufacturing workflow. So it's kind of one of the newer service offerings from Aldevron around gene editing. We can actually incorporate this nuclease along with the guide into formulated RNP and then release that at GMP. So really just trying to ease this process of doing genome editing and make it as convenient as possible to kind of source the reagents and get to the important part, which is, of course, treating patients. So, that's just a quick overview of some of the protein tools that Aldevron has in the portfolio around genome editing, around CRISPR. Of course, there are some other technologies that Aldevron has brought into the fold. I've talked a lot about knockout. There's, of course, great interest in knock-in the context of CRISPR genome editing. And so, I'd love to throw this over to my colleague, Jim Williams, who is going to talk a little bit more about Nanoplasmid and some of the great applications there. So, Jim?
Great. Thank you for the introduction. I'm Jim Williams, Chief Scientific Officer at Nature Technology Corporation. NTC is now part of Aldevron. And so what I'm going to talk about is NTC's Nanoplasmid platform. This platform is used to improve the safety and performance of a wide range of gene and cell therapy vector types. So this is a poll question we're going to ask of the audience now. What you would think in terms of where you might use nanoplasmids to improve your program. And I'm going to go through more of the details later. But yeah, so take a few seconds to think about where you could improve your vector backbone for these different applications. Okay, so specifically, I'm going to give you a background on the nanoplasmid platform and how it's used to improve performance of a variety of gene and cell therapy products. Then I'm going to focus on its application in as a template for CRISPR, a nuclease mediated gene editing, mainly as a homology directed repair template that can be used to knock in things like cars into the genome. So first, I'm going to give you a bit of background on the nanoplasm platform and where it came from. So bacterial vectors contain a backbone that has to provide the replication and selection functions in the bacterial host. So the bacterial backbone is just strictly for those two functions, bacterial functions. So virtually everybody uses backbones that were originally designed in the 1980s. That would be the puck vectors and puck and PAMBL. They were all put together in the 80s, and they have two things. They have the puck origin, which is a replication origin that has a temperature sensitive replication origin. So in that case, you have a low copy at 30, and then when you do a heat induction to 37 to 42, the copy number of that replication origin is induced to a higher copy. And almost all the manufacturing processes out there for these puck vectors features a temperature shift, 30 to 37, 30 to 42, allowing you to produce the plasmid, um, allowing you to produce biomass with a low plasmid copy number, and then you induce the plasmid copy number prior to harvest and purification of the DNA. And one of the things with the puck origin is because it has this temperature sensitive origin, when you induce the copy number, you can go to a set copy number that's dictated by the strength of the primer promoter in the puck replication origin. So you have a set copy number you can get to. The other component is a selectable marker, and it's virtually always ampicillin or kanamycin. And where we came in was these backbones had not been really looked at since the 80s. And for any of you who are molecular biologists, work with AAV or lenti or transposon vectors, just take your backbone and blast it against a database, and you'll see it matches up with something that was put together in the 80s. These backbones are like 2.5, 3 kb large, and really until fairly recently, no one really looked at the functionality of the backbone. So we NTC focused on the backbone. And so, that your vector part, which is the payload region of the vector, virtually everyone focuses on that. Our focus was on the backbone. And we worked on the backbone in two different steps. The first thing was the selectable marker. There's a lot of concern from regulatory agencies about having selectable markers in gene and cell therapeutics. And so we were given the task by a number of clients to engineer out that selectable marker. But if you look at the concerns, so the concerns are numerous, right? You can get the selectable marker out into the environment. This has been shown in a lot of studies where the replication origin, the plasmid, gets out into the environment out of a production facility, and now you have that marker out in organisms downstream of a production plant. It can get into your endogenous flora in your intestine or through the skin delivery, or it can give you an untowards immune response, perhaps even an autoimmune response to that protein. So the objective really is not just to get rid of the antibiotic, but to get rid of any protein in the vector backbone. And there's not a lot of options out there. So we developed the RNA out system. RNA out is simply a short, about 150 base pair selectable marker that ticks all those boxes. It has no coding capacity, no homology to the human genome. It just has a 70 base pair bacterial expressed RNA called RNA out. And in the chromosome, we've engineered the strain to produce a target of that RNA out, which encodes a messenger RNA that has a Levan sucrase protein in it. So without a plasmid in it, the cell produces this Levan sucrase constitutively, and it is exported to the periplasm of the bacteria. If you add sucrose, That enzyme can catamerize sucrose into non-toxic levain, but not non-toxic to a bacteria. Gram-negative, that concatemers of levain interfere with cell wall synthesis, so the cells die. If you have RNA out, that short 70 base pair RNA hybridizes it to its target in that messenger RNA and prevents translation of the levain sucrase. So we end up with a selection that you basically do a transformation and then plate the cells on sucrose. If you don't have a plasmid, you have death. If you do have the plasmid, you prevent death by eliminating expression of that or translation of the levain sucrase. So that was the first component that was driven by regulatory desires to get rid of markers from the vectors. The second component was driven by the need to get around gene silencing. So this was actually work done by Marquet's lab at Stanford, where they had shown that vectors delivered to the liver that had a normal puck backbone, 2.5 kb backbone or smaller, much smaller, any of those backbones were silenced when they got expressed into the liver. So in other words, the vector shut off and you had no transgene expression. And their solution was to use a very short backbone called mini circles. So mini circles are produced by in vivo recombination in the bacteria to remove a large bacterial backbone and replace it with about 100 base pair mini circle backbone, which is from the product of the in vivo recombination. So the mini circle eliminated that transgene silencing, but mini circles are very difficult to translate clinically because they're so difficult to manufacture. They're very low yield, complex manufacturing. And so, what our goal was to get the benefits of a mini circle without actually having the difficulty manufacturing. So, we want something that was easy to manufacture, like a plasmid, but had the performance of a mini circle. So, we needed, therefore, to replace the puck origin with something much smaller to get this small backbone, less than 500 base pairs, which was what would we needed to get around the gene silencing. And so we replaced the puck origin with the R6K origin. And so R6K is a very short 300 base pair origin. So now our backbone is less than 500 base pairs. R6K, unlike puck, requires a trans produced replication protein for synthesis. So we had to put that on the chromosome. What we did was, because as I mentioned, all the standard puck plasmid manufacturing processes are heat inducible to go from a low to a high copy. We wired it such that the R6K rep protein was also heat producible. And that actually allowed us to drive the copy number higher because unlike puck, which has this intrinsic copy number that's set by the strength of its promoter, we can provide as much of the R6K rep protein in trans as we want since we're expressing it from the chromosome. So we actually managed to hit the target of actually increasing the copy number with the R6K origin. So increasing yields with these vectors, as well as we verified that we had got around the transgene silencing, and we had also got the obvious eliminating antibiotic markers. This backbone has no coding capacity, no homology to the human genome, no markers at all. In the process of doing this, though, we obtained two unexpected benefits. One was much higher transgene expression than we saw with plasmids or mini circles. And I think, it doesn't make sense scientifically. Why would be higher than mini circles? But I think it comes back to how difficult it is to make a mini circle. If you think about in vivo recombination, low copy vectors, it's difficult to make a pure product, get it properly supercoiled. So I think the lower performance of mini circles is probably just due to manufacturing difficulties, not not an advantage of 500 base pair backbone over 100 base pair backbone. And we verified this experimentally looking at 0 base pair backbones using MIP vector designs. And indeed, you don't see any further improvement. The second unexpected benefit was the reducing the transfection toxicity. At this time, people didn't realize how much transfection toxicity was occurring with puck vectors. Those large, untranscribed bacterial backbones, when they went into a cell, were causing all sorts of activation of various innate immunity pathways, probably recognized as incoming DNA virus and inactivated by heterochromatinization. And so you ended up having loss of expression and a lot of toxicity. And so those are the two main attributes of this platform that were unexpected. less vector inactivation, the higher expression, and then the less transfection associated toxicity. Those, along with getting rid of the antibiotic markers, are where we got the benefit across a wide range of vector platforms. So, I'll just walk through them. Gene therapy vectors. Well, obviously that increased expression level and duration is of value. With non-viral vectors, hitting the expression level that you need for efficacy is hard to do, and so having much higher expression really helps get to efficacy that you need for these non-viral vectors, as well as maybe reduced doses. In the area of cell therapy, both transposons and CRISPRs, that reducing transfection toxicity is a big deal. In both those types of applications, you take primary cells out of the patient and you nucleaffect, which is the current method of doing it, the DNA in. In the case of a plasmid, that causes a lot of cell death. In the case of a nanoplasmid or a mini circle, much less cell death. And that is hugely important because there's a viability threshold that regulatory agencies mandate to before you can put the cells back into the patient. So you really cut your manufacturing timelines there because you don't have to grow them as long to get the viability back, and because you didn't knock down viability as much, the cells are more viable. So the cell killing, in the case of like transposon vectors, been shown by Poseida studies, the cell killing is more effective. You have a better cell because you didn't kill it as much. And then because the vectors are not inactivated as much, you get enhanced gene editing efficacy, enhanced transposition rates. So you get more transposons or precise gene editing events in a less toxic backbone. In the area of lentivirus and AAV viruses, that's a little bit more complicated. Is When you think about that, when you do a retrofit of an AAV or a lentiviral vector, the virus you make is going to be exactly the same whether you use a plasmid or a nanoplasmid. So we're not changing anything of the virus, but we have different benefits there. Like with a lentiviral vector, you generally have 4 vectors that you're transfecting into cells, and only cells that get all 4 vectors transfected in and have the vectors actually expressed are the ones that are going to produce virus. So it's not a surprise when you have something that's less vector inactivation, less transfection as toxicity, that you get enhanced manufacturing viral titers when you retrofit lentiviral vectors to nanoplasmids because of the less silencing. AAV vectors, interesting, I list the primary drivers. So the AAV field has learned relatively recently that the packaging of the vector, the cis vector that has the ITRs in it, is not a perfect process, and that maybe about 98% of them are from the ITR to the ITR with your payload, but about 2% of the viruses are made with the reverse packaging of the backbone. And so this has recently been shown and has been driving a lot of people to look at nanoplasms for AAV, because you're transferring enormous number of viruses with marker to the patient, like 1 to 2% of a 10 to the 12th dose, for example, is a lot of viruses with the marker, and obviously regulatory concerns. And then, of course, we get the higher manufacturing yields with nanoplasms in general because we are able to drive the copy number from that chromosomally expressed rep protein. This is one more safety feature with the nanoplasmas. As I mentioned, the puck origin is promiscuous and can get out into the environment. So it's a concern that has been shown to actually be the case, certainly with getting out of production facilities, getting into the environmental organisms. So the nanoplasm, because the R6K origin has to have this engineered host strain that produces the rep protein for replication, this is a dead end. It's like a mini circle in the sense it cannot replicate in an endogenous organism. So you eliminate this transfer to the organisms. That's quite a regulatory concern. So let's just summarize. We feel like we've basically combined the best attributes of plasmid and mini circle vectors in the nanoplasmid platform. So on the bottom, you see we've gotten around that promiscuous replication of the puck origin and now have a strain restricted dead end vector is not going to get out in the environment. Then we've captured the high yield and scalable manufacture of a plasmid vector. We've addressed the antibiotic marker or any protein marker really with all vectors. We've gotten from the mini circle, eliminating the transgene transfection, and got lower transfection toxicity based on the smaller backbone. And then that unexpected high transgene expression that we got was, which is probably due to the small background, less vector inactivation, and the fact that we can make a high purity product. And just the right just illustrates that when I say high transfection transgene expression, it can be much dramatically higher than with a plasma. This is work done by a group at Old Dominion. A number of groups have actually published on use of nanoplasm for a variety of different applications. This one just highlights where they took a very optimized plasmid vector, the GWiz expression vector, and compared it to the nanoplasmid equivalent, just with the shorter backbone. And you can see much higher expression level with the nanoplasmid versus the GWiz in vivo um, in mice in this particular application. So I'm going to end by focusing a little bit on use of nanoplasms as CRISPR HDR templates, and you get a number of benefits there, as I highlight on the left. You're going to get improved gene editing and less transfection toxicity. And when you look at the alternatives for doing a knock-in, you can think about using a plasmid or a linear DNA. And the study on the right just shows in this particular group when they looked at nanoplasms versus linear DNA for gene knock-ins, they had much better results with nanoplasmids. And this study, this is a recent paper that came out. This is a work done by Genentech that fairly exhaustively looked at nanoplasmids versus plasmids versus linear DNA templates with a variety of different lengths of inserts, lengths of homology arms, and making doing knock-in into primary human T cells. So this is what an application, if you want to put a CAR or TCR into a human T cell, this is an application, that a very relevant study for looking at the benefit of the different types of vectors. And so this is a complicated slide. You can't really see much, but on the right, if you look at the right of the chart, the purple bars, there's the three different things. At the top, you have linear, and the far right is the number of edited cells that you got. The middle is plasmid, far right, number of edited cells, and then the bottom is nanoplasmid. And you can see that they basically consistently found that they got nearly twice the number of edited cells compared to a plasmid format, and in this particular study, 3 times compared with linear DNA. And I think this slide ties back into what we were talking about before, was what Max talked about at the outset. You see they actually use the Aldevron Cas9. So they're showing here using Aldevron reagents for both the CRISPR HDR template for knock-in, as well as the enzyme used to actually knock it in, that you get superior performance. So that's one of the things that NTC is very excited about, is now that we're part of Aldevron, we're part of a start to finish solution. We can provide the CRISPR template. We can also provide the backbone that'll help you improve the performance of a wide variety of vector platforms, as I went through, with direct gene transfer vectors like non-viral gene therapy vectors, DNA vaccination vectors, and then, of course, the transposon gene editing, viral vectors, poly-A vectors. I mean, one of the things I really didn't get into is with the area of AAV and poly-A messenger RNA vectors. Those ones contain structured DNA. AAV has a ITR structure. Poly-A has a long, 80 to 120 base pair poly-A repeat in it. And we've found that the R6K origin really replicates through those structures much more effectively than the puck origin. So then you combine that with a cell line we developed, a reviver cell line, to stabilize ITRs and poly-A repeats. You get a much higher manufacturing yield with these things, with greater stability than you would with a standard puck vector. So that's another driver for people in the messenger RNA and the AV field coming to us to do nanoplasmid retrofits. So this is the Aldevron advantage. We've always been at Nature Tech. We provided the front end, we provided the vector, we provided the cloning, we provided research grade. But now we're part of that full solution, that now you can go to your much larger scales at Aldevron. You can get GMP, and you can get, for gene editing, you can get the proteins that you need for knock-ins. And here's our second poll question. we're interested in working with you on nanoplasmids and other CRISPR reagents. And, we'd like to contact you if that will work out.
Jim, Max, some really great insights and information, not only about Aldevron services, but the Nanoplasmid platform as well. It looks really, really cool and interesting. And from the look of it, our audience already has a bunch of questions, so I think that they're interested in it as well. To address that, our Q&A is coming up in just a moment. So for all you audience members who might have a question for Max or Jim, don't hesitate to type your question into the Ask a Question box and hit submit. All right, with all that said, no more ado, let's get the Q&A started. Bear with us for just a moment as we make the transition into the Q&A session.
All right, everyone, thanks so much for joining us for the Q&A session. We have a bunch of questions here for Max and Jim, so let's get to them and try to get through as many questions as possible. Gentlemen, first question we have, an audience member would like to know, are there any advantages for the high expression levels from nanoplasmids in gene editing?
Yeah, so I think the way to look at that is that high expression is kind of a indicator that it hasn't been inactivated. So when you look at like a non-viral gene therapy, the high expression is that it shows that a lot of the vector that might have been inactivated by heterochromatinization of the backbone has now not been inactivated, and so you have high expression. So how that would translate to a transposon or a CRISPR therapy is that if it was inactivated, it's not available to be cleaved by the transposase or the Cas9. And so the high expression, although the expression isn't translating, the readout of higher integration, because more of the vector is available to be cleaved by the targeting enzymes, that would be the readout. It's not really higher expression, it's higher integration and frequency.
All righty, thank you for that, Jim. Next question we have, an audience member says, have you experienced size limitations to the sequence edited by Cas9? And is there any advantages of using SpyFi Cas9 combined with nanoplasmids?
I think this is kind of a hybrid question. Max, you want to handle like the first half?
Yeah, I mean, well, as it relates to sequences that you can target using SpyFi Cas9, it's really analogous to the sequences that you could target with a wild type ST-Cas9. So the GG-PAM, that's pretty much the limitation we're aware of in terms of targetability for our products.
Yeah, and then the other thing is that, the SpyFi Cas9 was using that Genentech study, and they went up to about 9 kb, right, with the size. So in terms of, a nanoplasmid, there really doesn't seem to be a size limit.
All righty, thanks for that, guys. Next question we have states, what would you recommend as the optimal DNA concentration for the most effective Cas9 performance with no influence on cell growth or viability? And do nanoplasmids show any improvement to the traditional plasmid?
Yeah, and so that if you look at the results from the table, you'll see that nanoplasmid will give you more integration at lower concentrations, which kind of facilitates getting around some of that toxicity that you get. I do think you're going to have to optimize, depending on your system, depending on your guide RNA, the concentration you use, but expect to be able to use considerably less with nanoplasmid to get a elevated number of integrins.
All right, thank you for that. Next question we have, this is a pretty good question here. An audience member would like to know, does Aldevron provide support to develop the nanoplasmid HDR templates, and can it be provided at GMP grade?
So the support, I'll let Max answer, but certainly the GMP grade, yes, numerous groups have, that's part of our whole excitement with NTC being acquired by Aldevron, is now we can have a start to finish solution. Aldevron has made numerous nanoplasmids GMP, so yeah, it's just a smooth transition to GMP.
Yeah, and I guess I could add to that. You know, if you're interested in transitioning over to a nanoplasmid template, you know, we'd certainly love to talk. You know, sit down with you and your team and talk through it. We can certainly help provide some guidance there. We're also quite familiar with transitioning from kind of traditional plasmid backbones into the nanoplasmid and doing that conversion.
All right, great. Next question. Are the reduced off-target effects observed through different cell types? If not, do you help in troubleshooting off-target effects using SpyFi Cas9?
Yeah, that's a great question. So we absolutely you can see the the reduction of off-target effects using SpyFi in multiple cell types. I think the data we showed today was some T cell data as well as IPS. The original paper that I referenced from IDT and Porteous Lab actually looked at T cells as well as hematopoietic stem cells, and we've heard anecdotally, certainly from other clients who are working in other systems, that it does have that reduction. In terms of troubleshooting off targets, we can help out where we can. We kind of like to say we're not doing the gene editing here at Aldevron, but we can certainly provide the tools. So, when it comes down to doing things like guide screening, there may be certain in vitro assays that we can help with as part of our R&P manufacturing process. But ultimately, we've found that our clients tend to have better tools kind of at their fingertips to look at the off targets in their editing workflow that are most relevant to them.
All righty, thank you for that, Max. Next question we have, this is, I think, a very important question for any molecular biologist out there. An audience member would like to know, can my existing construct or plasmid vector be turned into a nanoplasmid?
Yeah, we call that our retrofit service. That's like the primary way in which people get nanoplasmid vectors to test. I mean, sometimes people come to us and just have us make it synthetically from scratch, which is fine. But often they have existing plasmids that they send us, and that's just a standard thing. Ideally, you have restriction sites. It's got cloned into a vector that we can just excise it from your vector and put it in the nanoplasmid backbone. We can adapt it to any of those sites. If there's no sites, we can just PCR it. But yeah, that's the standard way you would get a nanoplasmid retrofit of your vector.
All right, thanks, Jim. Next question we have, do you expect that SpyFi Cas9 nuclease will have reduced off-target effects throughout different diseases?
Yeah, I would certainly hope so, right? And what we've seen so far, through 4 years of working with clients on this product at GMP in a variety of diseases, that it does have that effect. Ultimately, the SpyFi, what we've seen is that reduction of off targets is across a wide variety of genes, guides, loci. So, in that context, you would expect then that a wide variety of different diseases would be addressable. Pretty much anything that you would think about addressing with a SP-Cas9, with that benefit of the reduced off-target effects. We've seen it in a variety of cancers, as well as sickle cell have been some of the early programs, but we really think we're just scratching the surface in terms of what clients are doing with this product.
All righty, thanks for that, Max. Next question we have, an audience member would like to know, what are the major benefits of Nanoplasmid's design as an HDR template when used with the Cas9 nucleases?
So that would be, again, less toxicity when you transfect it into primary cells, and then less inactivation, so more of the DNA is available to be cleaved by Cas9, which translates to greater number of integrants into the genome. So less toxicity, more viable cell with more integrants.
All righty, thank you for that, Jim. Next question we have, an audience member asked, what support is available for clients to meet the changing regulatory requirements?
Max?
Yeah, I guess I can take this one. We have a regulatory team here at Aldevron that not only serves our kind of internal purposes to make sure that our operation is in compliance, but also that our clients
have the kind of support they need around our products and services for their filings, both with the FDA and international regulatory bodies. So additionally, I should say we have master files, drug master files available for the GMP SPI-5 Cas9 as well as our GMP wild-type Cas9. And that's been really, I think, helpful for groups that are filing INDs. It just really eases the process of submitting that drug substance section for those materials. And additionally, for things like the Nanoplasmid or, custom Cas or international filings where there is no master file available, our team is certainly ready and willing to, help draft up those relevant sections of that filing. So the idea is definitely to make it as easy as possible to do your filings today, and then also in the future, as things shift and solidify around cell and gene therapy.
All right, thanks for that, Max. Next question we have, this is a pretty important question, coming from Frank, who'd like to know, is there a size restriction of the inserted gene in the nanoplasmid?
Well, I think with, at least with CRISPR, I've seen inserts up to like 11 kb or so. So, I haven't gone, our clients or users haven't gone larger than that, but I don't think there's a limitation.
All right, thank you for that, Jim. And with that, it looks like we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the Gen website. That's at genenginews.com for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends and colleagues who you think might be interested in it. I'd like to thank Max and Jim for their very informative presentations, and I'd like to thank you, the audience, for your attention and very thoughtful questions, and a very special thanks to Aldevron for sponsoring this webinar. Hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy.